So thank you for joining us today. My name is Maura McGinty-Kloss, and I am the Director of Programs and Communications for the Natural Areas Association. And we're delighted that you're joining us to be a part of our 3-day native plant series. You may only be joining us for one or multiple programs, but we're glad you're here. So the purpose of NAA webinars is not just to convey information, but to engage natural areas professionals on topics of interest that highlight emerging science, methodologies, and best practices. And we encourage you to participate in this webinar by posing comments, questions, and chats. Sometimes some of the best materials are things that you share with all of us, as well as what the presenter is sharing. At the end of the presentation, we'll address as many questions in chat as we can. And I also always just like to invite all of you, as you're watching this webinar and if you think of another topic that you would really like to see presented, please don't hesitate to send us an email at info@naturalareas.org. We want to take a moment just to thank our sponsor, the United States Forest Service. They enable us to be able to offer a lot of programming and great quality science to as many people who work in natural areas as possible. And we just really appreciate their support. And I want to take just a couple of minutes for those of you who might be new to the Natural Areas Association. We're a national nonprofit organization. We serve practitioners who manage ecologically significant landscapes to protect biodiversity. Protecting natural areas in perpetuity requires quality science to inform practices and access to reliable resources. NAA provides science-based content for natural area practitioners and research who seek applied science, practical solutions, and knowledge transfer related to the management of their natural areas. We offer a suite of programs such as this webinar, symposia, lectures, lots of other things that are opportunities for you to engage with your peers and are convenient so that you can build them into your busy schedule. I just want to highlight NAA's flagship publication, which is the Natural Area Journal. It comes out 4 times a year: January, April, July, and October. July is already available. October will be coming out anytime. And the journal is a peer-reviewed publication. It's available through our website. Members automatically receive a subscription, but articles are available to anyone through Bio1. And we hope that you'll take part in some of the great research that's being published in the journal. Later this month, we are also offering a virtual symposium, Nature's Frontline: The Role of Natural Areas and Climate Resilience in the Central Appalachian Forests. This is from 11 a.m. to 4:45 p.m. Eastern Time. And this program is really important. And we are so excited to be able to bring together a group of experts who will consider the role of natural areas as a critical strategic component to mitigating impacts of human-caused climate disruption. We will examine the science and equip practitioners with the information needed to inform management plans, educate policymakers and decision makers, and enlighten the public on the vital role of natural areas in offsetting the impacts of climate change on our planet. We have a wonderful group of experts who have spend their days thinking about nothing but this and related topics, including Dr. Reed Noss, Dr. Mark Anderson, Dr. Christopher Williams, Abigail Weinberg, and Dr. Charles Lafon. And it will be facilitated by land management practitioner, Dr. Ryan Klotz of the Virginia Natural Heritage Program. So it's an all-star cast, and we'll hope you consider joining us for that day. We also are offering a State Natural Areas Program roundtable on the forestry and wildlife management for the benefit of biodiversity. It's November 9 from 12 noon to 2 p.m. And this is an opportunity for state program people who manage state program areas, but it's also for open to anyone else who wants to participate in the conversation. We are currently seeking case studies. So we would like to focus this how wildlife biologists and foresters are working with state, local, and federal agencies and non-government organizations to manage not more than just game and species and wood products, but a more holistic range of habitats. And if you're doing that work, please consider submitting a case study. Please reach out to me. You can email us at info@naturalareas.org. we would love to be able to feature, potentially feature your case in this program. And something new, we've never done this before, but we are launching the Innovators in Natural Area Science Lecture. It is to highlight thought leadership and new discoveries that contribute to the protection of ecologically significant landscapes. And our first is being hosted December 1. It's virtual, noon to 2:30 in the afternoon, Eastern time. And we're featuring Dr. Suzanne Simard, the author of Finding the Mother Tree, Discovering the Wisdom of the Forest. And again, we're very excited to be featuring Dr. Simard. The program will include a lecture from her, as well as an opportunity to exchange ideas through a panel or fireside chat through some sort of an interactive program element. So we hope you'll consider joining us for that. And now, the reason you're here, let's get to today's program. Thank you for your patience as I go through all of that. Let me introduce our speaker. So Wes Knapp is the Chief Botanist at NatureServe. He's a leading biodiversity conservation nonprofit in the United States. NatureServe leverages the power of science, data, and technology to guide biodiversity conservation and stewardship, and is certainly an important partner of the NAA. Wes has over 20 years of experience in the Natural Heritage Program Network as a botanist, ecologist with both Maryland, North Carolina Natural Heritage Programs. He has extensive field experience across much of the United States, with additional field work in Australia, Canada, and Central America. His research includes the first examination of the extinct plants of the United States and Canada, new plant species descriptions, and treatments of various plant groups in various floras and manuals. Wes has published his first book, Vascular Plants of Maryland, USA, a comprehensive account of the state's botanical diversity, which is freely available through the Smithsonian Scholarly Press. His research interests include identifying and preventing plant extinction events, describing undescribed plant species, systematics, ecology, and taxonomy. He has a BS from Catawba College, an MS from Delaware State University, and is a PhD student at the University of North Carolina at Chapel Hill in Alan Weekly's lab. So at this point, I'm going to turn it over to you, Wes.
All right, well, thank you all for having me. I've been a proponent of the natural areas and tending for a long time, and I'm glad to be back now that I'm at NatureServe. And I get to talk about one of my passion projects and probably the future direction of most of my research, which will be identifying and preventing plant extinction events. We're going to focus on the USA and Canada today, which is really why I'm here with a publication recently in conservation biology. This is open access, so the link will be shared with y'all. Now, I'm going to go through some background about this project, how I got there, why I did this work, but also I want to really quickly touch that this was not just me leading an initiative. This was a huge collaboration. You'll see the authorship there. Anne Francis is talking to y'all tomorrow. She's with USDA ARS, and then Reed Noss, the third author. He's been intertwined with natural areas for probably his entire career. So this is a wonderful group of people to talk about. But I want to first start out with my interest in extinct plants with some background, because I started as a field botanist in 2001. So I'm really dating myself now. And I got a job with the Maryland Heritage Program, and I had this job to all of a sudden understand the whole flora of the state and identify natural areas for conservation. But I was pretty unequipped for that. So I studied the rare plant list night and day to get better acquainted with my job. And this plant here really captured my imagination, Nuttall's my chrysanthemum, because it was the only plant in the state flora that was GH, globally historic, and X, extinct. And I didn't realize we had extinct plants in our backyards. It seems to be a problem of the tropics, not necessarily Maryland, USA. But I did everything I could in my power to relocate this plant. That included learning as much about it as I could, but unfortunately, spoiler alert, I didn't rediscover it because it's extinct. But this is it. This is Nuttall's my chrysanthemum. It is an intertidal mudflat plant. It is the opposite of charismatic megafauna. This is a very cryptic small species that was last observed on the 13th of September, 1941. There are multiple duplicates of this plant. And then it wasn't even thought to be rare. And from what I understand, the Nature Conservancy at the time had scorecard ratings for their heritage programs. And this one's one of those plants that everyone in the room looked at each other. What do you mean you don't know? You don't have this? Who's seen this? Anyone seen this? And it was already extinct. It's pretty sad, to be honest. But the idea that we had extinct plants in eastern United States surprised me, because when you learn about extinct plants, you really know about one, Franklinia altamaha, the Franklinia tree. And it's the one told about in botanical books and biology texts, and that it was collected a single time in the wild on October 1st, 1765 by Bartram. And his book, Bartram's Travels, which is on the shelf behind me, it's a pretty unexciting story. He talks about going down a trail, seeing this beautiful shrub, collecting it, and wanting to name it after Benjamin Franklin. And that's it. We don't really know why this species went extinct. There's a lot of hypotheses about it. And thankfully, it's in the horticultural trade. So this is technically called extinct in the wild. But it made me realize there's a big data gap in our understanding of what extinct plants were and why they went extinct. Because I'm like an action-based non-academic scientist. I want actionable results. So I wanted to figure out why things went extinct in the past so we can have meaningful action to prevent extinctions in the future. So we wanted to compile a list of all the extinct plants from the United States and Canada, calculate an extinction rate, because you might hear a lot that, we're in the Anthropocene now. We expect extinction rates to increase over time, but we don't really know what our baseline firm extinction rate is. We wanted to draw an extinction event is a conservation failure. So we wanted to draw attention to these conservation failures and learn what we can to prevent future extinction events. Before I get too deep into the methods, I want to first point out that there are two different categories of extinct I'll be lumping together today for simplicity. These are presumed extinct, which we call GX extinct, or GH, or globally historic. And I want everyone to remember that extinction is a hypothesis. There's no proving an extinction event. I hope we're wrong on every one of these. I hope every plant is rediscovered, but we can say that there have been no confirmed sightings of any of these plants we're going to talk about today with over 25 years. They have varying degrees of survey effort, which influences the declaration of extinct versus globally historical extinct or presumed extinct. And I'm not talking about extirpations. There are plenty of plants that are going to be still extant in Mexico or in Cuba that are now extirpated from the United States. That is not what we're talking about. We're talking about extinction events. That goes for Canada as well. There's plenty of plants extirpated from Canada that are still found in the United States. That's not what we're talking about here. And we're looking at extinction since European settlement, because that's when the naming system that we all use for plants came about. We really don't have much data before that to build upon. So the study area is completely artificial. I want to make that clear, but it does parallel the Flora of North America series. So we're talking about the Flora of North America, North Mexico, which is the United States and Canada. I'm not talking about bryophytes today, but there is an extinct bryophyte, this Neomacuenia nitida, which is extinct from Canada, interestingly. That is something that should be sought for, but we're not going to look at bryophytes today. And we did not include Central America because the flora is relatively poor. A lot still to do. And then also we didn't look at Hawaii or Puerto Rico because of issues of island biogeography. If you include Hawaii, which is the global epicenter of extinction events, the data I present today becomes very skewed. I wanted to look at the continental landmass in and of itself. So how do you possibly go about figuring out what plants are extinct? We did all the typical things you'd expect, which included lots of literature review, lots of hitting the books like that cartoon suggests, and a lot of examination of the primary literature. A lot of conversations with different botanists across the US and Canada about what's extinct in their areas. But then lastly, we're so fortunate that we have this major database, Biotics, that compiles all the data of these NatureServe member programs, which are natural heritage programs and Canadian data centers across the US and Canada. So we can look at these two categories, GH and GX, which I alluded to earlier. So if you're not familiar with NatureServe methodology, it's a very simple scale of 5 to 1. This is an extinction matrix with the most common species at the subnational level being S5, with the rarest at the subnational level being S1. You can do the same thing with your national rank, the N rank, or the global rank, the G rank. So we have two other categories we don't talk about a lot, thankfully. We have GH, globally historic, and GX, extinct. And that's what we'll be looking at today. And this is what we flagged as our preliminary source of data on top of the literature review to look at what plants could be extinct in the study area. But when you mine a database like Biotics, you have inherent problems with taxonomy. You have to make sure all the names are created equally. And that's hard to do. Because if you just look at the database, we had 150 taxa that were presumably extinct. But that ranged from things like Franklinia, which is universally accepted as a distinct taxonomic unit, or things like Rubus fryi, which is either a global single site endemic from West Virginia or it's not real and should be subsumed with Rubus alleghaniensis. Those are two very extreme options. And when you're talking about the limited conservation dollars we have to spend them as precisely as we can. So we came up with an independent way to vet scientific names. We call this the Index of Taxonomic Uncertainty, the ITU. And this is the scientific consensus of a name. We wanted to look at all the floras and manuals that struggled to tell us what the plants were in the flora. Those people who are writing keys and writing descriptions have to handle this every time they do a treatment. So we looked at the individual treatments to develop a score, the ITU. This was used using calculated using novel taxonomic treatments, not just padding the same expert who did numerous treatments. That would count as one for that author. So we looked at distinct, unique treatments. And we did not use this calculating from databases like NatureServe's database or BONAP or USDA plants. We went right to the primary sources, those people writing floras and manuals, to see how they felt an entity should be recognized. So our grading system is much like the United States grade school system, which ranges A through F. A, as a plant with an ITU of A, is a unanimously and universally recognized entity like Franklinia altamaha. Very rarely Franklinia is put in a different genus, but is always recognized as a distinct species, Franklinia altamaha. We have plants like Juncus purvidus, which have a majority of taxonomic acceptance. This is almost always recognized as an extinct plant from Massachusetts, but there is one treatment that considered it a waif from Europe. So the taxonomic understanding, there is really a consensus that this is a unique and distinct entity from Massachusetts. So that's a score of B. We have plants that meet the score of C, which is minority taxonomic acceptance like Narthekium montanum. Now, this has more recently come into vogue to recognize as distinct, and there are numerous treatments that recognize this as distinct from the northeastern Narthekium americanum, but more often than not, it is lumped. So this gets a category of C because there's a lot of debate around this entity. Then we have categories D and F. These are very taxonomically dubious. Things like that Rubus fryeii that I mentioned before, or plants like Pennell's Penstemon leptanthus, which was described new to science and then never recognized by anyone else. So that's a score of F. So for this work, we looked at the ITU scores of A, B, and C, which gives us 65 taxa presumed extinct in the United States and Canada. There's one plant that has a NA, so there, no ITU score, because it was most recently recognized as distinct. We couldn't calculate a value. I'll talk about that one specifically later on. But as a botanist who spends a lot of time looking at different treatments and manuals, I was surprised at how many A's there were. It's surprising to see that much consensus about what entities exist. And graphically, here's just a quick visual guide to show you have this big batch of A's, a big batch of D and F, and then a small smattering of B's and C's that are recognized. Now, any name on this list may or may not be real. I think categories like D and F need a lot of work. They could be extinctions that have long been unrecognized or dark extinctions, which we'll talk about in the future. But we had to have somewhere to start, and this is where we started with categories A, B, and C. So some of the results that we had 51 species extinct, 65 total taxa, 42 taxa, or 64%, were single site endemics. They were plants known from only one place on Earth. And that matches an analysis done of global plant extinctions, which showed that extremely narrow geographic distributions are the most likely indicator of an extinction event. Now, the question we have to ask, and we'll talk about this a little later in the future, is were these plants really single-site endemics, or were they just collected from one place? Because one collection may not have been the totality of the species range. It may have just been what it was, where it was collected, so an artifact of sampling, if you will. I'll just point out briefly that only 2 of these plants were listed as extinct by the IUCN Red List, but we're working with them to update some of their listing. And then as for a baseline for the Anthropocene, there's about 15,882 species in the USA and Canada. So that's an extinction rate of about 0.4% of the flora. And since 1804, which is 216 years when we did the paper, there's about 0.3 taxa per year. So we're losing about 3 plants a decade. And we based that information on the year after the last observance of Franklinia, which was seen in 1803. So some of the more results, big families lost a lot of diversity like you'd expect, like the Asteraceae, the Fabaceae, the Rose family, the Bromes. Those are some of the biggest families on the planet. So we had 8, 7, 7, 6 extinctions respectively. But we have this sampling bias, or what we think is sampling bias, with other big families like the grasses and the sedges. Botanists avoid these groups. They're hyperdiverse. They can be cryptic. And the fact that we only have 2 grasses and 1 sedge is likely an artifact of biodiversity sampling. We have 1 orchid that's gone extinct. This was most likely an artifact of overcollection. We have 53 different genera. The largest genera were the hawthorns at 4, astragalus at 3, and then these 2 that you may not be familiar with here from the eastern United States, Cryptantha and Plagiobothris. So first I'll point out that the hawthorns are a very diverse group of plants with a lot of taxonomic uncertainty among these organisms. A lot of work still needs to be done on our understanding of what taxa exist in the hawthorns. So that 4 may be a very conservative estimate of the overall biodiversity lost in that genus. But I'll point out really quickly that these 3 Cryptantha and 3 Plagiobothris, these are from the same family, the Boraginaceae, and they are both subtribe Amiscaninae members, and these are sister genera, which is very surprising to have the 3rd or 4th largest family of extinction loss be completely represented in 2 sister genera. This argues, though we can't prove, there may have been some kind of pathogen that hit a number of species because of how closely related these are. That's a working hypothesis from that. So let's look at a map really briefly to look at where these extinction events were. You'll see that there is a very heavy southwestern bias to this map. This is most likely a twofold reason. The eastern United States was largely settled by European settlers before it was ever botanically inventoried. There is no way to tell how many extinctions happened before species were described in the East. In the West, at least we have some preliminary data from like the Lewis and Clark expeditions or early settlers collecting specimens, describing plants before the habitats were completely and wholesalely transformed. So there is a sampling bias to this map, we believe. A bit surprising, there's nothing in the upper Midwest, but this isn't an area of real biodiverse hotspot or endemism. And then there are 2 areas in Florida which are a bit surprising that there aren't extinct plants known from the Apalachicola and the Lake Wales Ridge. It's probably a good thing we don't have anything documented, but again, probably an artifact of settlement before exploration. I'll just point out also briefly, we had 2 species that had multi-state distribution. Most other species can be mapped to single or small site ranges. California was the biggest loser, what you'd expect. It has the largest flora in the United States. So by extension, you'd expect it to have the most extinctions. They had 19 taxa and a very broad coverage north to south, east to west in the state. Followed up by Texas. Everything's bigger in Texas except the extinction numbers. They only have 9, which is great, but it is the 2nd most diverse state floristically, so this makes sense. We have a largely southern distribution along the Mexican border, and hopefully some of these are still extant in Mexico, but none of the data argues that to this day. New Mexico and Florida are 3rd place right now with 4 taxa per state. But I'll talk about New Mexico very briefly because it shows the challenges of working with some of these extremely rare plant specimens. And the Helianthus prematurus, which is the lost sunflower, is really unmappable at the state level. It was attributed to New Mexico during the description of the species, but the data argues that the expedition where the plant was collected was actually in Arizona. So we're not really sure what state the species occurs in, but it's morphologically distinct and routinely recognized as a distinct species. Florida, again, a bit surprising in that there are 4 extinction events, but none from the Apalachicola or Lake Wales Ridge. They're all from southern Florida, but this does not bode well for the future of extinction events in Florida. Right now, we have 4 extinction events, but NAS et al. 2014 predict up to 29 Florida endemic plant taxa, which are under 2 meters of elevation, will become extinct over the next 100 years. That includes 6 Florida key endemics. So we expect to see Florida move from 4 extinction events that we know today to a total of 34 in 100 years. Now, this is an opportunity to do something to prevent extinction events, and we'll talk about that a little more in the future, because plants that are in the wake of sea level rise have few options to them. So we had 4 extinction events in Florida, and surprisingly, we have 5 in New England, which again, probably an artifact of sampling and data availability. But I'll talk first about the smooth, slender crabgrass, in that this was a single site endemic from Massachusetts, which is a repeating theme that 64% of the extinct plants were single site endemics. This is known from a very impressive ridge cliff talus system inside of, I believe it's Manchester, New Hampshire. It was described by Fernald in 1920 from thin humus and shallow depressions. It has an ITU of B, so it's almost universally recognized as a distinct species. What I want to point out is that over collecting most likely led to the species extinction. Fernald himself collected 31 herbarium sheets, much like this one. Each sheet usually has more than one plant on it. Each sheet has numerous rooting specimens. It's highly unlikely that any species of this narrow distribution could support that level of collection at a site. So there's 38 total collections of the single site endemic from a very narrow, very small patch site in Manchester, New Hampshire. But that's not the only time Fernald unfortunately wreaked a little havoc. Juncus pervitus, another species he described, which has an ITU of B. This is sometimes considered a waif from Europe, but the Europeans first pointed out that that is very unlikely. A single site endemic to Hyannis, Massachusetts, which is on the Cape. This plant, so far we've discovered 56 herbarium sheets that Fernald made of this species. I'll point out that it has a very thick, coarse rhizome. So he was doing serious damage by collecting these stooling, firm rhizomes from below the soil. And that in his description of this species, he talks about how it's one single giant patch of a plant. So he most likely unintentionally may have caused helped lead to the extinction of this species. But this leads to an area of interest of mine, which is the documentation of dark extinctions. So I've alluded to dark extinctions in that they're species that went extinct before they were described new to science. So since 1994, 4 species have been described or recognized as distinct only based on old herbarium specimens. I predict that more dark extinctions will be discovered as we examine our collections more finely. And here are the 4 here. We have Coriospermum pallidum from the Pacific Northwest, Govinia floridana, which is an orchid from Florida, which is probably overcollected to extinction. And there's a photograph of it, I believe from 1974 or 64. Marshallia grandiflora, which we'll talk about here in just a second, and then Potentilla uliginosa, which is from California. So these dark extinction events are happening or being documented from across the country, which leads me to a dark extinction event where I helped shed light on. And this is the large flowered Barbara's buttons, Marshallia grandiflora. This is a project I worked with Derek Poindexter and Alan Weekly on at UNC Chapel Hill. And Marshallia grandiflora is, a beautiful plant. It has this awkward, kind of strange bimodal distribution up here in Pennsylvania, West Virginia, and then Kentucky, Tennessee. And when I moved to North Carolina, I studied that rare plant list like I did back in Maryland. And I was really surprised to see these 2 orange counties here, which means it was historical in North Carolina, because this is a species of high flowing raging river systems, like a scour species, if you will. So I pulled up the specimens to see where this plant came from, because that's tip 1 in rediscovering the plant, is know where it used to be found. And I was really blown away by the morphology of the plants from North Carolina. The plants in North Carolina are much larger, much more robust, large heads, large leaves, compared to the plants from Pennsylvania, which I was familiar with having, because this is a G3 plant. It's something I'd want to see, and I encountered a few times in my career as a botanist in the Mid-Atlantic. So this immediately set off some alarm bells that further investigation was needed. So I reached out to Derek and Alan, my collaborators, and they'd never examined this material, which led to this study. After we got some material, we did some analyses, and you can see the cypsali, or the fruits and the heads, are very different between the Marshallia from North Carolina and the Marshallia grandiflora from the rest of its range. And then this is Marshallia legrandii, most recently recognized species endemic to just 2 sites in North Carolina and Virginia, which argues that you can have very narrow endemics in this genus. We did some statistics. I won't spend a lot of time on this, just to show that we had 3 morphologically discontinuous groups in these plants. And what we have now is 3 recognized taxa, 3 species. We have Marshallia grandiflora, which was endemic to Western North Carolina and hasn't been seen in 100 years. We have what we're now naming Marshallia pulchra to replace the name grandiflora for the plants that occurred from Pennsylvania to Tennessee. And then Marshallia legrandii, that 2-county endemic I previously mentioned, which will be shown here. So the naming was the issue at hand here, is that the type Marshallia grandiflora applies to the North Carolina plants. So we had to rename the rest of the range. This was a dark extinction in that we had a name of an extinct plant being applied to a more widespread and extant species. So this is the stuff we now call Marshallia pulchra, which the type comes from my colleagues in the Western Pennsylvania Conservancy. We have the type of grandiflora here, so pulchra grandiflora, and this is legrandii, a narrow-leaved diabase gladiatus species. So that's just one of the dark extinctions that's been identified recently. Another exciting area that I didn't know a lot about when I began this extinction work was this concept of extinct in the wild. So when we say extinct, we mean no naturally occurring populations. And that's an important definition with some of the plants on this list. So in the paper, we document that there are 5 EW plants and then 2 EW question mark plants. And we put the question mark there to be very clear. These species, Crataegus delawarensis and Euonymus etropurpureus variety Cheetami, have been attributed to be living collections at gardens in Europe, but these gardens have not provided the necessary information to accurately identify these species. I doubt that they're accurately identified at this point, but I want to present them with question marks so the community can further vet these. I don't want to dismiss those. But this brings up the fascinating story of Arctostaphylos franciscana, the St. Francis Menzelia. This was thought to be extinct before one of my collaborators, Dan Glausenkamp, rediscovered this along Doylestown Road in 2007 or 2008. The challenge here is that this road is going to be expanded and developed. This was quickly going to be now extinct again. So we did as a community, what we could do best is we dug this up and moved it. Now, because we dug it up and moved it, we planted it. This is essentially a gardened individual. There's no naturally occurring populations. So that meets our definition of extinct in the wild. So we have 5 extinct in the wild plants in the paper, but I'm here to report, unfortunately, that we've now discovered a 6th extinct in the wild plant. This is Stophanamaria malarensis. This was endemic to a single site in Oregon. It is listed under the Endangered Species Act as endangered. A lot of resources have been put into preventing the extinction of this species, a lot of ex situ conservation, a lot of seed propagation, planted 4,500 seeds on site and watered them. 46,000 seeds were directly sowed, but unfortunately, there are no naturally occurring populations found. All these plants failed. And this is an example of most likely a very recently evolved species that co-occurs with its common progenitor, and there may be genetic issues here with the chromosome numbers, we think. But this leads to a more important story about extinction in gardens. What are we going to do with all these extinct in the wild plants? And this is a collaboration with Mount Cuba Center, US Bot Garden, Morton Arboretum, Portland State University, where we're trying to bring together all these extinct in the wild plants as a display. This may be a traveling display to put people in touch with extinct plants. These are our black rhinos. Humans need to know of our impact, feel and touch it. We hope to have signs for all these species. So this is Ron Lance here, noted Crataegus expert who's done some really great work to graft these few extinct Crataegus onto other material for distribution. That includes, that's where the Morton came in, as they had the last living plant of Crataegus fecunda, which they mailed to Ron. Ron has propagated some plants, and I got those to the Mount Cuba Center. And then this here on the left is a living collection of Arctostaphylos franciscana, which was mailed from California. So Mount Cuba now has 4 of the 6 extinct in the wild plants. We hope to round out that collection later this year or early next year to have all 6 for our display. So let's talk about some of the causes of extinction. Habitat destruction and alteration is clearly the leading cause. That's anthropogenic causes. But I'll bring up again that 42 extinct species, or 64%, were what we call single site endemics. And it's hard to know the reason of an extinction event unless it was a single site endemic that has been destroyed. All other times, you're kind of inferring something else happened. Oh, like Franklinia, for instance. Was it a problem of fire regime change? Was it a pathogen? We don't know. For the broad-ranging species like Crataegus fecunda and the micranthemum, what captured my imagination, it's most likely the death by 1,000 cuts. We had rip-wrapping of shorelines, conversion of habitats. We have invasive species invading these habitats. So the death by 1,000 cuts, so to speak. So there is a call to action here. This isn't all doom and gloom, but preventing extinction is the lowest bar for conservation success we can set, and we're not always preventing extinctions. We need to learn from these extinction events because they are conservation failures. And one of the things we're trying to do now with the collaboration of the US Botanic Garden is prioritize single site endemics for ex situ and in situ conservation. Now, there is a real need to prioritize here. I know we are mostly in situ, like on the ground conservation practitioners. Largely, we do a fantastic job, but we need to close the gap, so to speak, with our ex situ conservation practitioner friends and colleagues. So for instance, this is the Botanical Gardens Conservation International database. And if you just type in Sarracenia aureophila, this beautiful carnivorous plant, you'll see here at the bottom that there are 44 ex situ collections worldwide in a partnering institution for BGCI. 44. That is most likely more than the total number of extant naturally occurring populations. This one's done. I can understand why there's so much attention here. It's charismatic. It tells a story. People are infatuated. But what about something like this? This is Packer serpenticola. This is a single site endemic to North Carolina. yet there are 0 XU2 conservation collections. There is a liability here when we have these endemics, these narrow endemics not backed up offsite. So the next part of this talk will be about the need to prioritize single site endemics. And I have to switch the language a tiny bit because we all have an idea of what we mean when we say single site endemic in our head. It's a species that evolved at a single place. It'll most likely only be in that single place forever for various reasons, maybe geological. But we have plants that fall into a slightly different category that are just as imperiled, if not more. So in this instance here, we have 3 red dots, which represent populations with a 9-kilometer separation distance, which would clearly argue they're distinct populations using natural heritage methodology as our standard is 1 kilometer. But let's say we have a loss of 2 of these populations down to 1. This would not be a single site endemic, but it's just known from 1 occurrence or 1 known occurrence. And that's what this project is really trying to find, are these plants of 1 known occurrence. Now, I do get questions sometimes, is it already too late for these plants of 1 known occurrence? There could be genetic bottlenecks. It could have a lack of genetic diversity. The truth is we don't know. That may well be the case for some of these, but I would hate to sit on our hands getting an answer for that and then watch the extinction of an organism that we could have prevented. Then we also need to think about future techniques. If we have organisms off-site conserved, like seed banks, living collections, we have tools and options. And the tools we have today are going to be greater in 20 years. So why not just get these things site protected off-site? And this is a way of prioritizing within G1. The G1 category has 1,234 plants in the United States and Canada. Conservation dollars are few, so we have to spend them in as precise a way as possible. And this is one of my long-term goals at NatureServe, is to bring the ex-situ and in-situ conservation community more closely aligned if we could. So, in-situ conservation, this is all the really important stuff we do in natural areas, site management, site protection, purchase, prescribed fire, invasive species control, making sure the long-term viability of these species continues on the land. The ex-situ partners, where we don't always work as closely as we maybe should, that's where we do seed storage, that's where we do living collection, we do off-site protection of genetic diversity is what we're doing with ex-situ conservation. And I also like to point out, what good is a seed bank if the seed isn't viable? They test for that. What good is the seed if we don't know how to grow it? That's why we have living collections. And frankly, where these 2 little bubbles overlap, that is where the magic happens. And we should be doing a lot more in this magic zone. So part of this project is not only identify those plants of 1 known occurrence, but to disseminate that in a careful way to the community for prioritized action. Now let's look at a global single site endemic. This is a plant that evolved in one place and will ever be in one place. This is Arenaria livermorensis, known in the Davis Mountains of West Texas. Beautiful little cryptic species. But then we have plants like Ancestorosarphus kellii, which is a plant of 1 known occurrence. There used to be 2 occurrences. The easternmost is most likely extirpated, so we're down to 1. So this argues for the need of conservation action. So the data I'm going to present here is preliminary. We hope to be wrapping this up this winter, but this is a map of the map of biodiversity importance, which NatureServe created using that natural heritage type data, showing like a biodiversity hotspot map of the US. This is just plants in G1 through G3. So at risk or imperiled would be the term there. So an imperiled plant hotspot map. And the single site endemics are plants of 1 known occurrence, and you overlay them are where you'd almost expect them to be. Like California has a lot, and I'm showing the asterisks because any number with an asterisk is we're still vetting that data, so there could be some error there. So you have 29 from California, 11 from Utah, 12 from Texas, and then a smattering across the United States and other places. Like you might be surprised to hear that glaciated areas, Alaska, Vermont, New Jersey, have single site endemic plants or plants of 1 known occurrence. In those instances, they're single site endemics. But this, to me, is a prioritization of where we need to spend some resources geographically. So how are we doing? Frankly, not perfect, and we need to improve. So if you look at the 89 single site endemics we've vetted so far, only 9 of them are listed under the Endangered Species Act. According to US policy, extreme rarity does not equate to listing. So something like Solidago plumosa, the Yadkin River goldenrod, which is found in 1 site in North Carolina along the Yadkin River, was petitioned for federal listing and denied. So that this number will never be 89, but it should maybe be higher than 9. We'll have to dig into that a little more. The Center for Plant Conservation National Collection, which is a leading ex-situ partner, has 38 of these already captured. And then BGCI and their partnering gardens have 55 captured. There's still some work to do with CPC and BGCI in vetting the taxonomy of their organisms to make sure they're capturing the organisms we believe they are, to make sure the IDs are certain. But we have gaps to close here for sure. So I'm going to talk about two single-site endemics. One I already mentioned, because these are continued work projects that are yielding some really interesting results. So in the 2020 paper, we said that Digitaria filiformis variety levi-glumis was GH. We wanted to do more survey work before we moved it to the extinct category or rediscovered the plant. But we also had this strange question about what taxonomic rank should this plant have and what is the distribution of variety levi-glumis? Because right before our paper published in Conservation Biology, Digitaria filiformis variety levi-glumis, the single-site endemic from New Hampshire, was attributed to Mexico and Venezuela. That does not make a lot of biogeographic sense to me, but we needed to put an asterisk next to it, investigate it further, and that's what this next collaboration really entailed. Bill Nichols of the New Hampshire Natural Heritage Program led this and has done a wonderful job with Joseph Wipf, the Flora of North America author, Aaron Siegel at the University of New Hampshire Herbarium, Jorge Gabriel Sanchez-Ken, who's with the Mexicans, who was the Mexican official who discovered the plant and attributed it to Mexico. And then Craig Barrett in his lab with Cameron and Lauren. Those are undergrads who are doing some phylogenetic analysis for us. This is really exciting work. Anytime I can pull in like an international collaboration and bring in students. So we had the first question of what rank should apply to all of these entities, because there are 4 recognized varieties in the flora of North America that Wipf did his treatment on in 2003. And also, what's the right rank of this plant? Is it GH, is it GX, or is it extant and still in Mexico and Venezuela? So the first part is the fun part. That's the field work. And unfortunately, could not discover this species. And some of you may be keen-eyed and recognize that's Bill Brumback of New England fame, a botanist up there. They went out there years ago and they did another round of surveys recently, could not find Digitaria levi-glumis. But what they did find were 2 non-native crabgrasses that are invading this site. So you may have had the first overcollection by Fernald, where he collected that 31 specimens, and then a secondary invasion of non-native species into these habitats. So we are now moving this to extirpated from the site and extinct from New Hampshire. But we did a molecular analysis looking at all these varieties of the filiformis complex with some very interesting results. First, I'll point out that variety levi-glumis, all the collections sister together, and they sister next to the Florida, South Florida endemic Digitaria filiformis variety Dolichiophila. And then sister to those two are the Mexican plants we were able to sample. So this is very interesting, and Wipf is going to elevate all of these to the species level. So we'll have the extinct levi-glumis, the globally rare, this needs investigation, Digitaria Dolichiophila, and then the Mexican plants Dr. Wipf is going to recognize and describe himself, because there are numerous morphologic and molecular differences between the two. So we're going to have numerous new species elevated to species rank. We are moving levi-glumis from GH to GX. But there is a little bit of a question in what to do with the plants from Venezuela. We could not sample these destructively for our analysis, but Dr. Wipf was able to look at this herbarium sheet and definitively say that is not levi-glumis. This is most likely another distinct and undescribed Digitaria species. And I'll leave on like some good news, because extinct plants can be pretty depressing to think about and talk about. And this is the most recent thing on the most cutting edge information on Texas red oaks. And this is a big collaboration. I need to call out everybody here because credit should be given where credit is due. And that Ray Mims of US Bot Garden and Murphy Westwood are really the people responsible for this effort. But then we had a large field crew of John Santilli, Botanical Gardens, Adam Black with Bartlett Tree Services, Michael Easton, like really Mr. Texas Flora of the San Antonio Botanic Gardens, Emily Griswold, a noted oak expert from UC Davis, Philip Schultze from Lady Bird Wildflower Center, Elizabeth Thomas from Poly Hill, and Zara Wiley. These are all field people who've spent a lot of time in Southwest Texas, in particular Big Bend, and an effort I'll talk about in a minute. So why are we looking at the oaks of Southwest Texas? In that delimiting species can be really difficult in Southwest Texas. If you can't delimit the species in the field, it's hard to apply a conservation rank that's meaningful. You have to think about what we do. What is it? Where is it? And how is it doing? But if you can't tell the organism apart from another organism, then your decisions about what to conserve and how rare something is, or its extinction risk, can be wrong. And that's what this effort really stems down to, is species concept work. And this has led to 2 collection trips in Texas back in May and August of 2021, and then a most recent collection trip here in 2022. And I'll just point out some people. This is our ridiculous Botany Boys glamour shot. That's me, of course. This is Adam Black from Bartlett, Michael Eason from San Antonio. And this is Dr. Andrew Hipp, who's doing the molecular work for us. Same crew here in this photo, but this is also Marianne Renault. She is a journalist who embedded with us in 2021 when we did our survey work. And she wrote a couple really nice stories in the New Republic on the hunt for Quercus tardifolia, which is really what this expedition stemmed to do, is work on species level delimitations and then potentially rediscover an extinct oak species. So why are these oaks so difficult? Here's a plate from the flora of North America that I kind of pieced together and then stole parts of from Dr. Andrew Hipp. But you get the idea in that there's a smear of morphologies here. These are not easy to identify in the field, and many of them are very rare. So from left to right, we have Quercus graciliformis, which is a G1, only known from a narrow area of Southwest Texas, including Big Bend. Quercus amorii, which is G5, secure, abundant, common. Quercus robusta, which is another G1 plant, I believe endemic to Big Bend. Quercus tardifolia, which is believed to be extinct, GH. And then Quercus gravesii, another ubiquitous, common, secure species found throughout Southwest Texas. So as you can tell, there's a lot of variation in the leaf morphology, and this happens in Texas, especially with moisture. If you have a wet year versus a dry year, the leaves on an individual tree can change dramatically. But you have to have an idea what the species are to rediscover them. And we spent some time at Sul Ross State University examining Quercus tardifolia specimens. This is the last known collection from 2011, collected by Dr. Mike Powell. And what I'll point out is that among all the oaks in Southwest Texas, this is very unique of the bristle-leaved species, in that you have this broad leaf. It's very thick, coriaceous leaf, and the underside is just covered with this indumentum, almost like a white arachnoid pubescent all over the undersides of the leaf. So of all the species in Southwest Texas, this one really quite jumps out as being distinct. And this is the one of the types. This is the holotype collected in Boot Canyon in 1939. Sorry about the date cut off there. It says an ITU of B, and that is almost always recognized as distinct. Sometimes it's considered a hybrid of unknown or uncertain parentage. But we are, for the molecular component of this project, we were able to sample Dr. Powell's collections and one of the isotypes housed in the California Academy of Sciences. So we get to compare what the genetic makeup was of this extinct plant compared to extant individuals in that group of species. But I want to shine some light on some of the fun stuff and exciting stuff. And that back in May, we did a week-long expedition to Big Bend National Park. The whole crew of us that I listed off there, 9 of us, I believe it was. That involved taking pack mules, multi-hours up these mountains, way deep back into Boot Canyon. And here's Boot Canyon. It gets the name from this kind of upside-down geological spire that kind of looks like a boot if you tilt your head the right way. But this is a hard place to get to and a hard place to survey. Back in 2021, we surveyed Boot Canyon, looked at the last known location of Quercus tardifolia, could not find it. But I noted that there was a trail running through this canyon, and there was a spring head there, as well as a cabin nearby. So this is a highly visited area, and it left a lot of other habitat that should be surveyed, which made me feel good about the GH rank, because GH indicates there's some likelihood of a species rediscovery, whereas X would mean truly gone. But this site, this is a rugged area. There's not much flat out there. These are incised in places, dry, crunchy, bouldery, hot, but they are a bit of sky islands that as you come up the mountain gradient, you get forest development and more moisture. And I'll never forget that a group of us spread out like a party looking for a lost child, went walking through the woods, and Michael Eason called me out, said, "Wes, you need to get over here." And when I came over, he had found this tree. This is the last known tree of Quercus tardifolia. And when I walked up, it was immediately distinct, and it matched the herbarium specimens with that deep pubescence on the undersurface and the fat leaves. From the earth looking up, you could see that the light wasn't penetrating through the leaves like it did the other 1,000 or plus oaks that we'd seen collectively throughout the trip. So it had a real gestalt difference about it morphologically. So we all gathered around for a celebratory selfie, but Adam Black, the guy here on the left, quickly pointed out this tree is, quote, not long for this world. Adam knows a lot about pathogens and tree health, and he pointed out that this area had been burned recently, so the stump was blackened. There was a fungal infection. And if you're keen-eyed enough, you'll notice this kind of bushy, shaggy look, or lots of epicormic branches coming right out of the stem. So that's a sign of poor health. So now what are we going to do about this? Now we're working with the National Park Service and Bartlett and Adams San Antonio Botanical Gardens to do some off-site conservation, taking a lot of cuttings, grafting them onto other oak species, get these off-site and get them growing. If there are any acorns produced, we really can't trust their genetics. Oaks can be very promiscuous, so we wouldn't know the parentage of any acorns on this tree. But we'll also point out we only found one. This was day 3 of a 7-day trip. We all hope to find more after that. This is the one that we found. Special shout out to Kelsey here, who is our camp manager, because we hiked 10 plus miles a day, and it was always nice to have dinner ready for you, not to deal with the food and the logistics. So future work, lots to do here, that we need to complete our one known occurrence plant identification progress, and then get that into the hands of practitioners like you and professionals in the ex-situ community. We need to make sure that these sites are protected, so they're not going to be developed. What I mean by protection, that the invasive species are under control, what other management needs are needed, because all our eggs are in one basket. If we don't manage for this species correctly, we could lose it. And we have to work about the ex-situ component, the off-site component, the seed storage, the living gardens. This is where it's going to be a bit of a challenge, how to disseminate that data, because if a plant is a single site endemic described from one place in the world, figuring out that one place will be easy from the literature. So we just have to figure out how to disseminate that data in an accurate and morally acceptable way. Hopefully that will happen this winter or early next spring. So in conclusion, there is just so much left to do. Extinction is more common than previously thought. We've documented 65, definitely more in the dark extinctions arena, probably more still to come. And then there's always a lag time in our identification of extinction event, because you can't prove an extinction event. So we expect this number to go up as we dig more and more into the data. So it's 51 species and 14 infraspecific taxa. Those global single site endemics and plants of one known occurrence need to be prioritized for conservation action. That includes in situ and ex situ. Our two worlds need to come together here to prevent extinctions, because things are slipping through the cracks, unfortunately. We all just need to work more collaboratively, because frankly, the stakes are way too high. And I am going to take a minute and actually acknowledge these people who are really fundamental to this work, because this is not the Wesley Knapp show. So the U.S. Arboretum, the U.S. Herbarium, and the Smithsonian Library, namely Rusty Russell and Robin Everly, just let me set up shop and go through the literature back when I was working with the Maryland Heritage Program. I need to thank the NatureServe Network programs. I was a member with Maryland and North Carolina for 20 years. And if it wasn't for that data set, we would really have almost no clue about what plants are extinct and what is on the precipice of extinction. Gary Moore of the USDA Plants Database was a collaborator on the Extinct Plants Project, and he helped provide the open access charges so everyone can get this data. Ron Lance, wonderful Crataegus expert. He literally wrote the book Haws on Crataegus. I recommend getting a copy. Abby Meyer and Nick Barton at the Botanical Gardens Conservation International were really helpful in mining their mega data set, and they'll probably be collaborators on the one known occurrence plant project. Matt Lobdell was very patient in taking the photographs of Crataegus fecunda at the precise moment it is identifiable, the species. And if it wasn't for those photos, Ron never could have provided an identification of an extinct in the wild plant. Marianne, I already mentioned her. She is just wonderful. I first met her with a publication through the New York Times on extinct plants, and then she's followed up with a couple just beautifully written stories in the New Republic about Quercus tardifolia. There's some other articles there by Susan Millis at Science News you can check out. Matt Candidis, I've been on his podcast twice. I'll give those a listen. And then here at NatureServe, a lot of our funding comes from federal partners to rank the flora. So Peggy Allwell and David Pivarunas, big thumbs up and shout outs for all the work they do to help plant conservation. Here's my information and my contact. I will also point out, I will leave you with the late Jim Fowler's wonderful photo of Trillium georgiatum, a single site endemic only known from one place in Georgia. Thank you, and I look forward to your questions.
Well, thank you. That was amazing. And we've got about 5 minutes for questions. We do have a few. I think I'm going to ask the one that I think you could probably answer the quickest. Just curious if you knew of any extinct species that had specialized relationships with pollinators that would also be endangered.
We can only speculate at this point. So little is known about these extinct plants. Heck, you go outside, we don't know a lot about most of our rare flora, what pollinates them. I agree there had to be some linkages there, especially with our single site endemics. But right now, I don't even want to venture a guess.
The other one that's quick, and then the others are kind of related to an issue I think that comes up for you a lot. This question is: how big an impact has been altered disturbance? How big an impact has been altered disturbance regimes, i.e., lack of fire?
Yeah, huge, absolutely massive. Fire is the most preeminent but missing component of our landscape. But again, it's hard to quantify all these things, and that's why I hesitate to put meaningful percentages on what impacted a species or what caused a species extinction. But it's one of the death by 1,000 cuts for sure. It may have been what caused Franklinia to go extinct, but that's the wonderful example in that we just truly don't know. We're speculating, but we know fire was a ubiquitous, important component of our landscape. So it most likely was an impactful piece.
So there's a couple questions, and they're really about translocation. One talks about as conservation botanists, we need to establish criteria for decision making because it's going to happen. We're going to need to translocate, but is there a criteria? And do you know of anyone who might be doing that?
Yeah, this is something we're going to have to deal with. Whoever wrote that, you are spot on. I agree. Like that example from Florida, I didn't want to get into it with the limited time we had, but that we're either looking at massive extinction events or we're going to have to move stuff. And we probably have a moral obligation to move these on a prioritized way because we're causing their extinction. There are criteria out there, like Emily Coffey at the Atlanta Botanical Garden has been working a lot in this arena. There's a lot published here. And this is another way I want to meaningfully help and lead conservation with the Natural Heritage Program Network and the ex-situ gardens community, is to talk about those criteria. There are published criteria out there on when and where. But and there are even differences in assisted migration and translocation, but it's something we're going to have to deal with or let happen kind of in the background because it's happening already in an unregulated, inconsistent way. And this should be handled in a very scientific and careful manner.
So what is your view on conducting reintroductions of state listed plants as a way to help prevent extirpations at the local level? I understand that it's widely discouraged to do so for federally listed species without an experimental design approach, and state natural heritage programs appear to have adopted the same point of view for state listed species. I agree that we should always prioritize managing and protecting plants where they naturally occur, but I wonder whether reintroductions of state listed plants should be attempted, especially for those that may be listed as an artifact of drawing arbitrary political boundaries where the species habitat is restricted to a small fraction of the entire state. Did you follow?
Yeah, so localized extirpations are the first step towards an extinction event in range collapse. They can lead to an extinction event. So they should be prevented whenever possible. This yields a future project to look at plants of one known occurrence within states for priority action, because they should be prioritized, because again, you have no redundant collections. Each species needs to be vetted individually. So I resist like a blanket statement about reintroduction of plants. So it would have to be, what species are we specifically talking about? Where are we talking about a reintroduction? What benefit does this gain? I think there is, meaningful conversation to have here. I wouldn't be wholesale opposed to it, but it needs to be carefully considered on a case-by-case basis and get the conservation leaders from your state together to agree upon these activities. The worst thing we can do is to move stuff haphazardly or without documentation, because I've seen that in my own career. I've discovered what I thought was an amazing discovery to find out that it was planted. Well, thankfully, we knew it was planted, but if I couldn't figure that out, that site would have been a conservation priority. We may have purchased it. We may have essentially wasted our resources on a garden. And that's not what we want either. So we need to be very careful anytime we move plants, but I'm not opposed to it under the right circumstances.
Okay, we are at 1 o'clock, but we have one more question that I'll give to you. Do you consider that ex situ not successful due to not considering soil interactions such as microbes, hemiparasitics, and specialist pollinators?
So is that about the Stephanomiria that I talked about that went extinct despite ex situ efforts? If so, I think that what's happened there, and I'm relying on the experts in the Pacific Northwest to really tell me, is that this looks like it was a very recent speciation through a diploid event. So we have the common progenitor there and the doubling of chromosomes in the same site. And that's not necessarily conducive to long-term species viability. Let's be honest, extinction's a natural process. Almost every species that's ever evolved has gone extinct over time. And this may have been a species that evolved through this diploid event and would have naturally gone extinct anyways. And that's why it's important to think about what we're spending our resources on. I'm glad they spent the resources on it. It's now ex situ. We can grow it in gardens and tell a story or put it back someday. I can't imagine some microbial change in that instance, but I can see that being an important thing to consider for other species if you want to reintroduce them to different sites.
Okay. Well, on that note, thank you so much. This was fascinating. We appreciated all the great information that you provided. I did add the links to the resources and articles that you mentioned into chat, and those will also be available when we send you the recording. So everyone who registered for the webinar will receive a recording and links to all the resources. So Wes, thank you again. We appreciate it, and we hope to see you again soon.
Thanks.
Okay, bye-bye.